ADP becomes ATP. We add that phosphate bond onto ATP, generating the currency we now need to do work. This process is oxidative phosphorylation. Because the generation of ATP is the result of the proton motive force. That's what we're doing up here when we generate our hydrogen gradient. This is the proton motive force, and our ATP is being generated by oxidative phosphorylation. So this is for eukaryotes. This is for what's going on in mitochondria. Bacteria are a little bit different. They don't have the fourth complex, but they still establish a proton gradient. But they can do a little bit more with their proton gradient than we can. Some prokaryotes have proteins embedded in the membrane that, when they allow the hydrogen ions to flow through, so when the hydrogen ion passes through, they also bring substrate with them. So it's a way of moving things into the cell. They can use these protein channels now, along with the hydrogen gradient that's been established, and as hydrogen ions move into the cell, a substrate can piggyback along and move into the cell as well. In addition to that, as hydrogen ions move into the cell, the bacteria can use it to move their flagella. Now, they've put numbers on what it takes to generate power here. With regards to ATP, about 10 hydrogen ions would generate about 3 ATP as it moves through. It only takes 1 hydrogen ion per 1 molecule of substrate generally to pass through the channel. But to cause the flagella to move, 2,100 hydrogen ions have to pass through the gate in order to move the flagella. So a lot more required to move flagella.
Now, is that the way they always move the flagella, or just when they always move it that way? Probably.
I would say no. They may use ATP on occasion, and again, it may vary depending on microbe. I don't know, to be honest with you.
So prokaryotes are the only one that's bringing the hydrogen and substrate together into the cell.
Yeah, they use that. They use the proton motive force to move things into the cell. Yes. Eukaryotes don't do that. Now, a couple of things we can mention here. With regards to this process, if oxygen is not there, okay, so your cells aren't getting oxygen, what happens then is that this complex cannot get rid of the electrons that it has, which means that the quinone here, or in this case it's cytochrome C, that's passing the electrons it has can't get rid of its electrons, which means this complex can't get rid of its electrons, which means this quinone can't get rid of its electrons, which means this can't get rid of its electrons, which means ultimately this can't get rid of its electrons. When that happens, when your NADH molecules cannot get rid of their electrons, you don't generate any NAD, and you need NAD to receive the electrons that are over here, the electrons that are giving off here, the electrons that are being given off there. Without oxygen, the whole system shuts down. Without something to give those electrons to, you don't recycle the NAD molecule, and therefore you don't have electron carriers to pass those electrons to. So the TCA cycle doesn't function. And the glycolysis pathway would not function except for one process. Fermentation. What fermentation does is it takes this pyruvate molecule that we've generated here and it uses it as a terminal electron acceptor. Over here, where am I? Oxygen is our terminal electron acceptor. Oxygen is accepting the electron at the end of the process. If it's not there, we can use pyruvate as an electron acceptor. So basically, pyruvate will take the electron from NADH, so regenerating our electron carrier that now can find its way back to the glycolysis cycle, so at least glycolysis can continue. The rest of the stuff won't continue, but glycolysis can. And what do we produce in return? Lactic acid. Lactic acid is the product of pyruvate serving as that electron acceptor. Lactic acid is what builds up in your muscles when you go anaerobic in your exercise. Some microbes, there's another step in here I'm leaving out, will convert pyruvate into alcohol. So fermentation produces two possible, well, there's several possible we'll talk about next time, but two of the possible end products we've already talked about, lactic acid and alcohol, result of fermentation, basically just taking the pyruvate molecule that's generated from glycolysis and adding the electron to it. But in doing so, that means that then we will generate an NADH molecule that will allow for glycolysis to continue. So if we think about this for a minute, we get a little bit of energy from glycolysis, 2 ATP. We get a lot more from the TCA cycle through the proton motive force. About a total of 36 ATP generated for every glucose molecule. Fermentation, not nearly so much. Only 2 ATP generated from fermentation. So cells don't get nearly as much energy from the fermentation process. Okay, I have a little video for you to watch. I don't have a connection. This is going to be a bummer. Somebody know what I'm doing wrong, or do I just not have a connection? What's that?
If you click on the icon, there's
I don't want to do anything.
There's a thing on the bottom right. Check the